Hello everybody, welcome to the Advanced Modeling Principles series. This is the lecture that will be focused on GIS model, also known as Geographic Information System model. My name is Luke Qian and I'm a postdoc at Food Safety Lab from Cornell University. Before diving into the content, let's first look at the learning outcomes for this lecture. I hope upon the completion of this course, students should be able to explain the importance of farm-level factors on produce safety, and the students should be able to explain what is GIS and how GIS works. Next, students should be able to explain different steps in developing a GIS or geospatial model, and also explain the practical applications of using GIS model in produce safety. Lastly, the students should be able to describe recent research projects using GIS models for predicting foodborne pathogens in agricultural water or in other applications. This lecture will be divided into 4 parts. At part 1, we will talk about a few of recent foodborne outbreaks associated with fresh produce due to contamination at pre-harvest environment. This is to help us understand why controlling pathogen at pre-harvest level is important. In part 2, we'll talk about Geographic Information System, also known as GIS, understanding what is component and what are the typical data processing techniques used in GIS. This will help us understand how we can use the input from GIS to build our model later. At part 3, we'll dive into a practical application in a case study. In this case study, we'll be talking about a model that was built to predict the prevalence of Listeria monocytogenes in New York State produce fields. As the last part, we'll expand from this case study to discuss other potential and future applications of GIS for produce safety and food safety, including using GIS models for monitoring pathogens in agricultural water. Now, let's begin with the first part, which is produce outbreaks due to contamination at pre-harvest environment. To start, let's first discuss why we need to employ geographic information model tools towards produce safety. As we can see from this graph, in recent years we have learned that fresh produce, including leafy greens contaminated with foodborne pathogens, represents a major contributor to foodborne illness. In fact, between 1998 and 2008, approximately 46% of the estimated foodborne illnesses in the US were attributed to fresh produce, which was nearly double the number of estimated foodborne illnesses that were associated with other food commodity categories, including meat, poultry, dairy, and egg, and fish and shellfish. Therefore, increasingly, fresh produce has been portrayed as a concern for public health. In addition, a summary of investigated foodborne illness outbreak data in the US between 2004 and 2013 found that, second only to multi-ingredient foods, fresh produce was a single-ingredient food vehicle responsible for the most outbreak and foodborne illness cases. This highlights the need to controlling pathogen and foodborne illnesses in the sector of fresh produce throughout the supply chain. Given the complex nature of fresh produce supply chain and absence of a definitive kill step such as heating that is applied to fresh produce along this fork-to-fork continuum, there are opportunities for pathogens to contaminate fresh produce prior to consumption. As an example, at post-harvest levels, pathogens can contaminate fresh produce if the truck that's delivering the crates that contain the fresh produce are contaminated. In addition, at facilities, during the cooling, washing, and packing processes, the equipment can harbor pathogens and will cross-contaminate the fresh produce. At retail and food services, if an employee are not following the required guidelines, they can also contain pathogens that will transfer into fresh produce. Although this contamination can occur at both the pre-harvest and post-harvest stages of supply chain, as this lecture is going to focus on the geospatial model that will be applied at the pre-harvest level, we will focus more on the pre-harvest factors that will cause foodborne pathogen contamination. Applying geospatial model at the pre-harvest level can help us better understand risks and develop strategies to mitigate fresh produce contamination at the pre-harvest stage of the supply chain, which can occur from inputs such as agricultural water, soil, and manure used as biological soil amendments, livestock runoff from adjacent lands, and wildlife intrusion. It is important to highlight that many of these same agricultural inputs have been suspected as sources of pathogen contamination in several previous fresh produce outbreaks. For example, manure was suspected as a contamination vehicle in a 2014 Salmonella outbreak linked to cucumber. Wildlife intrusion was suspected as a vehicle of contamination in 2014 Shiga toxin-producing E. coli outbreak linked to strawberry in the US. In addition, wildlife intrusion was also suspected as a vehicle in 2004 Yersinia outbreak linked to carrots in Finland. In addition, agricultural water have been suspected in multiple foodborne outbreaks. For example, in 2018, E. coli 0157H7 outbreak was linked to romaine lettuce due to agricultural water contamination. In 2008, Salmonella outbreak linked to peppers, and in 2005, Salmonella outbreak linked to tomatoes were both linked to agricultural water as a contamination source. And in some outbreaks, multiple potential pre-harvest contamination sources were identified. For example, in a 2006 E. coli 0157H7 outbreak linked to spinach, samples of cow manure, wild boar feces, and creek water close to the growing area were tested positive for the outbreak strain, highlighting the potential for multiple pre-harvest sources to contribute to foodborne illnesses outbreak risks in fresh produce. Therefore, managing and monitoring only one pre-harvest factors is not enough. We have to take a systematic look at all factors in order to manage the foodborne pathogen at the pre-harvest level. Here is a table of a more comprehensive list of foodborne disease outbreaks linked to pre-harvest contamination of produce up until 2014. As we can see, these cases are not alone, and these contamination sources are very typical for pre-harvest level contamination. The likelihood of foodborne pathogens being present in various pre-harvest contamination sources can be influenced by a variety of highly complex environmental landscape factors, including the geographic location of the growing region and its proximity to adjacent roads, water sources, and other agricultural farms, as well as weather factors such as temperature, humidity, wind speed, precipitation, all of which can influence things like soil moisture and flooding from proximal water sources. Given this increasing complexity, we need to consider lots of data inputs when trying to model ways to mitigate foodborne pathogen contamination of fresh produce. In part 2, we'll talk more in detail about how these type of data inputs can be generated and analyzed using geographic information system. So what exactly is a geographic information system? By definition, GIS is a computer system for capturing, storing, tracking, and displaying data related to positions on Earth's surface. The quote from National Geographic mentioned that GIS enables people to more easily see, analyze, and understand patterns, relationships, and trends between different datasets. GIS works by uploading multiple data files, displaying them, visualizing, and stacking them on top of each other, so that the users can examine the ways in which datasets relate to each other. Layers can contain a variety of spatial and non-spatial attributes such as geographic locations and land ownership. Building tools will allow users to join datasets, alter visualization based on data attributes, and perform basic geostatistic analysis. As a data management software, GIS can also store the output from other software and provide input for statistical analysis. As a result, GIS is highly flexible and scalable from micro to macro level. In addition, GIS is also powerful because it is a relational database that facilitates data management, use, and analysis. For example, we can merge data from multiple fields collected at one time point. In relationship to food microbiology and produce safety, we can collect microbial data, for example, pathogen presence and different time points and specific locations, molecular data, field collected environmental data, such as water attributes, and management practice data, such as the movement of harvesters or other equipment or employees. In addition, we can also collect real-time data from public sources, for example, weather data and remote sensing data. We can also use outputs from statistical models as input for GIS management system. For example, if we have a sub-model that can predict animal intrusion in specific locations, we can use that as an input for our GIS system. With relation to produce safety specifically, GIS tool can be used to generate hotspot maps that will show areas where a pathogen was repeatedly isolated or odds of isolation is high. Let's imagine a grower that has multiple produce samples tested positive for El mono in a produce field and wanted to perform root cause analysis to determine why. In this case, one could develop a hotspot map to identify areas with higher or lower density of positive samples. It was the figure on the right-hand side was done in 2013, in which a spinach field was sampled. Samples were tested positive for El mono and were used to generate a hotspot map. This is similar to a kernel density map and shows areas with more cases as a warmer color. However, doing this at one time point will only give us part of the story. To enhance the use, we can generate multiple times from different time series and looking at them sequentially to find out more important messages. In part 3, we will showcase a case study in which a GIS model was developed to predict the prevalence of El mono in New York State produce fields. Using this example, we can more concretely understand what GIS application can achieve. This geospatial model was developed in 2011. As a case study, 20 New York State produce fields were sampled between 2009 to 2011. Drag swabs, soil, water, and fecal samples were collected and tested for Listeria pathogenes. In addition, GPS coordinates were recorded for each sample. Meteorological and spatial data were also collected for each time point and sampling point. Data from multiple sources were then merged into one dataset using GIS. These GIS data were used to create input for GIS model in R. The model that was selected was called Classification and Regression Tree, also known as CART. The CART algorithm is structured as a sequence of questions, the answers to which determine what the next question, if any, should be. The result of this question is a tree-like structure where the ends are terminal nodes at which point there are no more questions. As we can see from the right-hand side figure, this is an example of CART. The main elements of CART are rules for splitting data at the nodes based on the value of one variable. For example, the rule for splitting data at the first node is based on the water rule. And if the value is less than 37.5 meters from water, then the sample will be classified into the right-hand side. In addition, another element of CARD is a stopping rule for deciding when a branch is terminal and can be split no more, which is the last rule as we see as the pastoral rule. And finally, a prediction for the target variable in each terminal node. As I mentioned earlier, at each terminal node, the sample will be classified into different outputs. In summary, a card is a classification model, and in general, classification models were used to create homogeneous classes for each level of a target categorical variable using a series of hierarchical rules. In our case, these hierarchical rules are based on meteorological and geospatial factors, and the factor level we want to create for these classes are high and low prevalence of Listeria monocytogenes. Let's use a more easily understandable concept to illustrate how classification regression tree works. Imagine if you want to divide a set of red and blue shapes into three homogeneous groups. You may figure out the biggest difference within this group is the shape. Depending on the shape, whether it is triangular or circle, we can first split by shape into two large groups. Then, based on the size of the shape, we can further split the triangles into large triangles and small triangles, so these subgroups are more homogeneous. Similarly, as pathogen prevalence was not uniform in the pre-harvest environment, this means that the risks of contamination in different locations of field or farm are not equal. We can use this CART algorithm to figure out which environmental factors may lead to a higher predicted prevalence. As you can see from this CART, water rule will be the first deciding rule for the splitting. If the sample is closer to the water, it has more likelihood of being contaminated with Listeria. Then we would move on to the impervious cover rules. If it is near IC, then we would assume it has higher prevalence of Listeria. Following on, we have available water storage rule and pasture rules, which will further make our subgroups more homogenized. At the end, we can easily see by dividing the samples based on different meteorological and geographic factors, we can achieve predicting the presence of pathogens in different locations of farm. Then we can further use GIS to visualize our model output. Using the results of the statistical analysis, a new map was made in GIS using the relevant data layers, including proximity to road, water, pasture, and available water storage. In combination of the risk levels, we can generate a map of our model output. For example, the map on the right-hand side is showing the risk levels of different areas in the farm by different color shade. The expected prevalence of Listeria monocytogenes is in parentheses in the legend. This map will allow us to better find high-risk areas in the farms and potentially apply intervention strategies to manage the pathogen prevalence in the future. To make sure our model is accurate and to make sure that our model results can be more generalized and applicable to future applications, we further conducted the model validation, during which our four farms were divided into high and low predictive prevalence areas as previously designated. Drag swaps were sampled from different subplots, and Listeria monocytogenes were tested for these samples. Using the validation input data and our previous developed model, we further categorized our model prediction into high prevalence sample and low prevalence sample. Although we can see that the prevalence level was a little different from previously. For example, in previous results, the high predicted risk from water rule will be roughly around 39%, but here it is lower than expected. We will still be able to see that water rules and pasture rules were two main factors that were able to predict Listeria prevalence. And with sample less than 37.5 meter from water, we've seen an odds ratio of 3 than the low predicted risk levels for the Listeria prevalence. Similarly, the odds ratio for the pastoral rule is 2.9. Therefore, these two factors are the main factors that are driving the model prediction. The previous application or case study we showed was conducted more than 10 years ago. Since then, GIS models have developed quite a bit. The extension of previous work has been leading to GIS applications in agricultural water and GIS models that increasingly use more types of data, such as water attributes. In this part 4, we'll talk potential and recent applications of GIS for produce and food safety. The findings of our previous GIS model can lead to the change of management plans for the produce farm. This can lead to risk prioritization and assessment. For example, our farms can target samples closer to water and take more samples from these sites in order to prevent potential contamination and take in-time actions. In addition, farms can potentially treat agricultural water if they feel certain locations are more likely to have water contamination. The farm can also track trends from year to year in case these contamination trends change due to reasons such as climate change. With increasingly collected data, the farm should have better ability to forecast food safety issues that occur in this farm. Overall, by collecting more data to conduct GIS models, the farm should be able to have a surveillance system in place that will continue to provide insights on the current or reoccurrence of these foodborne pathogens. The GIS application for producer safety is more and more promising because of agricultural and smartphone growers are increasingly more comfortable with GIS and related technologies. Farmers are increasingly use this type of technologies to predict the yield and economic benefits from their growing crops. Therefore, these systems are already in place to set an environment for monitoring food safety incidences. In addition, we have more and more open source software and publicly available data, such as remote sensing, weather conditions, and traffic conditions, among others. If there's free information out there, that means we can have more informed decisions to make using this information. If farmers can track trends and patterns within a growing region and year to year, they can better predict and address on-farm food safety issues. These principles have already been applied to other fields as well. For example, in the dairy sectors, weather conditions and farm management have been used to predict spoilage microorganisms in bulk tank milk. In recent years, spatial temporal models have been developed to predict when and where agricultural water is likely to be contaminated. This is showing another promising area of applying GIS model. This is a table that summarizes recent research publications on predicting presence of pathogens in agricultural water. In 2019, Pollard published a paper using pond samples to predict Salmonella presence in central Florida. In 2020, Weller published two papers that developed models to predict Salmonella and Listeria prevalence in watershed of Arizona and New York State, as well as streams in New York. In 2021, two papers were published to predict the prevalence and E. coli, as well as Salmonella and E. coli in Maryland and southwestern U.S. respectively. In 2022, a model was developed in Spain for predicting Salmonella in watershed. This collective body extended our understanding of different pathogen presence in different water bodies at different geographical locations. And together, they can increase our insights in understanding pathogen prevalence in water. Why is water important? Because surface water used for produce production has emerged as a primary source of pre-harvest microbial contamination of fresh produce. This is largely due to the fact that on-farm surface water can act as a reservoir and transmission pathway for foodborne pathogens. In fact, irrigation with untreated surface water has been repeatedly associated with isolation of key pathogens such as L. mono from pre-harvest environments. During irrigation, pathogens that are already present in a pre-harvest environment can bypass, via runoff and splash, physical barriers to movement into and within fields, resulting in produce contamination. Moreover, stochastic, low-level pre-harvest contamination can result in contamination of post-harvest environments such as packing houses, where pathogens can proliferate and contaminate large amounts of product. Thus, prevention of pre-harvest produce contamination, particularly by microbially contaminated surface water, is a critical concern for food safety and the produce industry. Recently, surface water has been implicated in multiple foodborne disease outbreaks linked to produce. In 2018, E. coli 0157H7 outbreak in romaine lettuce at Yuma, Arizona, and they found outbreak strains in irrigation canal. In addition, in 2024, Salmonella outbreak was identified and sourced back to cucumber grown in Florida. They also found outbreak strain in irrigation canal water. These two incidences provide evidence that canal water and irrigation water can be a major source of foodborne outbreaks. Therefore, monitoring when and where the surface water is contaminated is critical for maintaining the produce safety. Currently, the agricultural surface water is regulated by FSMA, which is known as Food Safety Modernization Act. This is the first federal attempt in the U.S. to regulate fresh produce safety. And the final rule on pre-harvest agricultural water that was published in July 2024 requires establishment of system-based pre-harvest agricultural water assessment for hazard identification and risk management decisions. This systematic approach is ideally can be powered by GIS-based models to identify when and where samples should be taken. This guidance, however, was not specific from the FSMA rules. When designing such a system, we need to evaluate multiple factors such as agricultural water system used, different practices, crop characteristics, and environmental factors. For example, the type of water distribution system, whether it is open or close to the environment, or the degree to which the system is protected from possible sources of contamination, including other uses of the water system, animal impacts, adjacent and nearby land use. Examples of agricultural water practices including the application methods such as overhead sprinkler versus spray, drip, furrow, flooding, and seepage irrigation. Example of crop characteristics is the susceptibility of covered produce to surface adhesion or internalization of hazard. So basically, different produce types are intrinsically having different risk levels. Example of environmental conditions including precipitation, air temperature, solar radiation, which were previously associated with pathogen prevalences. That concludes the end of this lecture. So the key takeaway from this lecture is that pre-harvest environment presents a major source of bacterial contamination in fresh produce. GIS is a powerful tool that can be used to integrate multiple data sources and provide predictors to develop GIS spatial models. It is also a great tool for visualize pathogen prevalence in different locations in the field or farm. Lastly, geospatial models such as classification and regression tree can help with risk management and assessment, as well as pathogen surveillance that will help us to continue managing the risk levels in our farms given different conditions. At the end, I want to acknowledge the funding source for this lecture. This research was supported by the United States Department of Agriculture, National Institute of Food and Agricultural Specialty Crop Research Initiative, Project Award number 2019-03139. The slides were generously provided by Dr. Daniel Weller. Thank you.